congratulations to the anniversary, and also IPK had anniversary this year. Due to the gene bank, we're a little bit older, so we are celebrating or celebrated already the 80th anniversary. But of course, the phenomics is way younger. So today I will present the whole plant installations, the pot-based phenotyping systems, and after me, I think that Astrid Juncker will present IPK Phenosphere installations, which is a worldwide unique infrastructure. And so the whole plant phenotyping systems, they give us a chance to assess the complexity of growth and the performance under the challenging conditions. And why that is important is basically we all know that we have an increasing world population. However, we have to feed them on less arable land in a changing climate that gives us more and more unfavorable conditions like heat, drought, but also diseases. While on the other hand, there is of course an increasing demand for quantity, but also for the quality of food. And this was illustrated here in the graphs below. So I hope you can see if I change to the next slide. So IPK has invested in phenomics since more than a decade now. And the levels reach from cellular up to the population level, and you find information on the infrastructure here. As said, I will now focus on the pot-based systems, and the new installations will be covered by Astrid. And just to remind us, so IPK has 3 of these pot-based systems. Of course, now this is bad because that's animated. So one is installed in a small phytochamber, and the 2 others are installed in semi-controlled greenhouses, and they are dedicated for different species sizes, but they all have basically the same composition. So they have the top and side view camera systems of the different types, so visual light, near-infrared light, and static fluorescence. And the small system and the very large system allow also phenotyping of several plants per carrier, depending on which kind of pot or tray you use. All of the system also, of course, have the automated watering, so you have full control of your watering scheme. What was updated then was that via DPPN, we also upgraded all of the system with kinetic chlorophyll fluorescence imaging to also get information, especially during stress, on photosynthetic performance. Moreover, in the large phenotyping platform, we have also upgraded to additional root phenotyping. That's illustrated here. So there is a so-called rhizopod where you have this front disc. This does not allow visible light to come through, but we are able to see the roots then via an NER sensitive camera. And this concept was already published earlier and was then applied to phenotype maize. And there is a new publication on the outcome of this root phenotyping using this rhizopod system here. But basically, I will not go into details here, but you can also nicely observe root growth over time additionally to the shoot growth data. The medium-sized system we have used since long to explore different drought scenarios, and we have started with the spring drought setup where we actually used IPK gene bank accession. So we had 100 single seed descent lines that were of course genotype. They were all of 2-rowed spring types. And at that time, we used 5 biological replicates per line and had 3 experiments per treatment, because at that time, nothing was really known about heritability, especially under stress in these systems. And we have published the GWAS data for both of the setups some years ago. Just to show you, and you're all aware of that, that you can illustrate nicely daily biomass growth in this system. So you can also model individual growth curves, for example, logistic modeling for the well water treatment, but also more complex growth models to look on the drought stress response. And we would get here from this biomass wilting curve in the spring drought setup that is followed by rewatering, we can, for each plant individually, assess the time where the growth is stopping, and we call that wilting time because afterwards our digital proxy for biomass is decreasing and then is recovering upon rewatering. And this was assessed for these GWAS panels, and we have first of all a high heritability that was then allowing us to reduce in further experiments the number of replicates or also experiment number for GWAS studies. And we could use this data, daily data, also for the temporal resolution of the QTLs, and this you see illustrated here. So this is always one graph represents one QTL with a log p value over time, and between the dashed line you have the drought period, before you have the establishment period, and then the recovery period. And in blue this is the well water treatment, and in orange you have the stress treatment. And then you find different type of QTLs. For example, you find QTL which are valid for the early growth stages, so they are significant in the seedling stage and then disappear over time. And you have other QTL which come up over time. And of course there is an influence of drought. So although we have constitutive QTL classified as QTL that we detect in both of the treatments, you see a significance shift. And also, for example, this QTL here on 7H was disappearing during the drought period and then reappearing in the recovery period, while the other most important QTL is in the frame of the PPDH1 flowering time gene in barley. And you see that it has more importance under drought treatment in the recovery phase than under well-watered conditions. And then, of course, interesting are the QTLs that appear only in the drought stress treatment. We found 4 of them, and they come up mainly in the time of severe stress and then disappear once the drought period ended. Also, you can find QTLs in this study, it was 1, that are relevant only for the recovery and also play no role under the well-watered conditions. And in this case, these were mainly in the vicinity of flowering time loci, and this is simply also the effect of phenology on biomass, but also then on drought stress response. What we also wanted to know is if this wilting time, as we call it, so the stop of growth and drought in this barley panel is occurring maybe at the same water status. And this you see here. So from the watering data, you can look on the watering or the water content calculated from the watering data. And this is, of course, in the drought period reducing very strongly. And then we cross this information with the red line. This is the average wilting time in the barley panel. Of course, there's variation for that. So yeah, some genotypes are more sensitive and start to wilt earlier, some are more tolerant. We also have a high heritability for this trait. And we did now for each plant check when is the wilting time and what's the water content at this time point, made statistics with that. Was also a highly heritable trait, and discovered that on average this is happening at 21% water content. However, it does not happen at the same time. There's also a huge range in the barley panel. And based on this threshold, we were then using this in further setups as a severe drought stress level that leads to wilting of plants. Later, we added with the fluorochem the photosynthesis data for this population, and we also see that photosynthesis in stress is strongly affected by this setup and is fast recovering. And interestingly, this change of or this drought responsive QTLs were mainly detected also in the time when the photosynthesis was already severely reduced. So I have to click here. Yeah, so with this information or experience, we went into a collaborative project with the Hebrew University by Shukri Sarangwa, and they have identified QTL for biomass and grain yield in the field coming from wild emmer, and introgressed these QTLs into Israeli red wheat, but also durum wheat cultivars. And we had then, for the first time ever, this material on our phenotyping system from day of sowing until really maturity. And we mimicked a drought scenario that should best match with the Israeli field. Drought development. So first you have no drought, then you start to develop drought. So we had a watering level for a mild drought period, and after flowering we intensified the drought by going to this threshold of 20%. And yeah, so of course now this is not animated. We were seeing, for example, for the 7A QTL in the Barnier background, that plants of the near isogenic line had in both conditions more biomass. They were not differing in phenology. They were also taller, and what was very nice to see with this setup, that based on the color value of the RGB images, you see also very nice the senescence phase, which is in well-watered conditions not different between the two lines. However, in the drought treatment, the near isogenic lines has a kind of stay green effect, so is slower senescing than the parent. Also, we saw, based on the biomass information, that the near isogenic line reacted later to the drought treatment. So plants from the two treatments started to differ later than in the parent compared to the near isogenic line. And we ended up with higher final biomass and also with higher final grain weight. So could verify the field or yield advantage even in our pot-based system. And we also saw a higher photosynthetic efficiency, and the plants also had a higher 1,000 kernel weight, and in the end also a higher water use efficiency. So the next point is that we are currently working on this effect of phenology. As said, so flowering time is affecting the biomass, and the starting biomass at the drought stress period is also connected with the wilting time. So plants with a higher biomass, of course, often start to wilt earlier, and you have to take that into account. And in the barley HEP population, NAM population, where white barley parents are crossed with Barke, a modern cultivar that is already well studied, and the phenology effect is well known here. We have now applied a setup where we start the drought for each plant individually at the same growth stage, which is BBCH 31, and then allow the plants to go down to this 20% field capacity, and then keep each plant for 14 days at this level, and then have again rewatering and observed the plants 14 more days in the recovery. And this is in collaboration, of course, with the Martin Luther University in Halle, and this data set is currently under exploitation. From wheat and barley, we moved into legumes in the frame of the INCREASE project. We have here now 200 diverse chickpea genotypes that have whole genome shotgun data, and my PhD student Madita is exploring this data at the moment. So we have conducted experiments for the whole population with the spring drought setup, have also information on photosynthesis, information, had added metabolomics during the drought stress phase, and also went to the final yield parameters. And she already has published from a common core set of 60 genotypes the effect of this spring drought setup on in these chickpea genotypes. And of course we see again a strong reaction to the drought in form of biomass. Although it's recovering, the gap to the well-water treatment is always big. And we see also an increase again in the yellow to green ratio as a stress indicator that is then, after rewatering, also slowly disappearing. Again, also a red to green ratio is increased. And we also see again a reduction, but also a fast recovery of the photosynthesis. And all these data are currently under analysis for the GWAS in chickpea. So these were just some examples how we use that systems for, in this examples for drought tolerance. We also performed heat stress experiments in wheat. And basically, high throughput phenotyping allows us to study really the dynamics of growth and the trait relationships during the life cycle. And we get high quality data, meaning high heritability, that allow also to phenotype with a low replicate number. And we can resolve this temporal dynamics of the genetic architecture, or even of complex traits, such as biomass, but also of drought tolerance related traits, also by modeling the growth curves. And these thresholds that we have developed also allowed us to phenotype different drought scenarios from short to long lasting, or at early stage or late stage. And with careful designs, you can also connect single plant growth to the field performance. Yeah, with that, I hope I kept the time, and also would need to acknowledge a lot of people, but I just want to highlight, of course, the research group at Heterosis, Thomas, the research group of image analysis, Evgeny Gladilin, the bioinformatic people under Uwe Scholz, and also the quantitative genetics group with Jochen Reif and the collaborators on the project. And thanks a lot for your patience, and I'm sorry it didn't work well with the presentation mode.
Thank you very much, Kerstin. Thank you for keeping the time.